Welcome to the Deep Dive. We're here to tackle complex topics and give you the core insights, basically a shortcut to getting up to speed. Today, we're talking about something fundamental, something happening inside you constantly cell signaling. It's this amazing, invisible language cells use to communicate with each other and with their world, from bacteria right up to us. And our source material, a really thorough chapter on cell signaling from molecular biology of the cell.
It really is fundamental. It governs just about everything in life. Think about how a single cell finds food, or how a complex organism like a human develops from one egg, or even just how our tissues keep themselves going, repairing things. It all comes down to cells sending and receiving messages. So yeah, this deep dive should give you those key pieces of information, maybe some surprising connections too.
Okay, let's start at the beginning then. Why bother? Why do cells even need to talk to each other? Seems complicated for tiny things.
Well, it's fascinating because even the simplest single-celled organisms are surprisingly social. Take bacteria. Many use something called quorum sensing. They release chemical signals, right? And when enough signal builds up, it tells them, hey, there's a crowd here. Then they can coordinate, maybe form a biofilm or produce antibiotics together. It's group action.
So even bacteria have committee meetings.
Huh? You could say that. And yeast cells, they secrete mating factors to find a partner and get ready to fuse. So even basic life depends on this kind of cooperation.
And in us, in multicellular organisms, it must be way more complex.
Oh, unbelievably complex. Our bodies are like these incredibly organized societies of cells. And communication is what holds it all together. Growth, division, cells becoming specialized tissues, it's all coordinated. And the basic setup is simple enough. You have signaling molecules, the messages, and receptor proteins, the receivers, that translate the message into action.
Okay, so how do they actually do it? Are there different ways to send a message, like depending on the distance?
Yes, exactly. In multicellular organisms, there are basically four main ways, and they differ in distance and speed. First, you have cells literally touching, that's contact-dependent signaling. Direct membrane contact, super important during development when cells are deciding what to become, and also for immune cells recognizing threats.
Like a cellular
A
handshake.
very specific one, yes. Then there's paracrine signaling. This is more local. Cells release signals, local mediators, into the fluid around them. These act on nearby neighbors. There's also autocrine signaling, where a cell signals itself.
Signals itself, why would it do that?
Well, sometimes it's for positive feedback, reinforcing a decision. But cancer cells often hijack it. They tell themselves to keep growing, which is, bad news.
Right. Okay, so that covers local chats. What about sending messages across the whole body?
For the long haul, you've got two main systems. Synaptic signaling, that's your nervous system. Neurons and electrical signals super fast, down long axons, then release neurotransmitters right onto a specific target cell at a synapse. Very fast, very targeted. And then there's endocrine signaling. This is the broadcast system. Endocrine cells dump hormones into the bloodstream. Those hormones travel everywhere, acting on any target cell in the body that has the right receptor. Think insulin, for example.
That makes sense. But hang on, if the bloodstream is full of hormones and the space between cells has local mediators, how does one cell pick out the right message? It sounds like signal overload.
That's a great question. It comes down to specificity. Each target cell has receptor proteins that are incredibly selective, like a lock and key. They bind their specific signal molecule very tightly with high affinity, even if it's only present in tiny concentrations. Most receptors are on the cell surface because most signals can't cross the membrane. But small fatty ones, like steroid hormones, they can slip through and bind to receptors inside the cell.
So the receptor is the key filter. And it's not just one signal hitting one receptor, is it?
No, absolutely not. A typical cell is swimming in hundreds of different signals. It has to integrate all that information. Often, a cell needs a specific combination of signals just to survive. Different combinations might tell it to grow, or divide, or become a specialized cell type. Without the right survival signals, many cells actually undergo programmed cell death, apoptosis.
Wow. So it's like needing multiple permissions to proceed.
Kind of, yeah. And what's really mind-bending is that the same exact signal molecule can trigger completely different responses in different types of cells. Acetylcholine is a perfect example. In heart pacemaker cells, it slows the heart down. In salivary glands, it triggers saliva secretion. Same receptor type even. But in skeletal muscle, it binds to a different type of receptor and causes contraction.
So the message is the same, but the interpretation is different.
Exactly. The cell's internal machinery, its history, its programming that determines the outcome, not just the signal itself.
Okay, this is getting really interesting. Let's talk about those receptors on the cell surface. What are the main types that actually start these internal conversations?
Right, so most cell surface receptors fall into three main families. First, ion channel coupled receptors. Think of them as little gates in the membrane. Signal binds, gate opens or closes, ions rush in or out. This changes the cell's electrical potential really fast. Crucial for nerve cells and muscle cells.
Got it. Quick electrical zap.
Pretty much. Second, G-protein coupled receptors, or GPCRs. These are huge. They don't act directly on an ion channel or enzyme. Instead, when the signal binds, the GPCR activates an intermediary, a helper protein called a G protein. This G protein then goes off and regulates other targets like enzymes or ion channels. It's an indirect relay.
Okay, a middleman. And the third type?
Enzyme-coupled receptors. These guys either are enzymes themselves or they're directly linked to enzymes they activate. Often they're protein kinases, enzymes that stick phosphate groups onto other proteins inside the cell, changing their activity.
Okay, so signal hits receptor. Then what? How does the message get amplified and processed inside?
Good question. That's where the intracellular signaling machinery takes over. You have second messengers. These are small molecules generated in large numbers when a receptor is activated. Things like cyclic AMP or calcium ions, which are water-soluble and diffuse to the cytosol, or diacylglycerol, which is lipid-soluble and stays in the membrane. They spread the signal quickly and amplify it because one activated receptor can lead to the production of many second messenger molecules.
Like a shout echoing down a hallway and getting louder.
Exactly. And besides these small messengers, you have lots of intracellular signaling proteins that act like molecular switches. They can be flipped from an inactive state to an active state and then back again.
How do they flip?
The most common way is by phosphorylation, adding a phosphate group, usually from ATP. Protein kinases are the enzymes that add phosphates, often turning a protein on. Protein phosphatases remove them, turning it off. A huge chunk of our proteins, maybe 30 to 50%, are regulated this way. It's a massive control system.
And the other type of switch?
The other main type involves GTP-binding proteins, or GTPases. These are on when they have GDP bound and off when they have GDP bound. They act like timers, too, because they slowly convert their own GTP to GDP, switching themselves off. And they have helper proteins. GEFs turn them on by helping them grab GTP, and GKPs turn them off faster by boosting that GTP to GDP conversion.
Okay, that sounds incredibly complex inside the cell, like a busy factory floor. How does anything stay specific? How do you stop signals meant for pathway A from accidentally triggering pathway B? All that crosstalk potential.
It's a real challenge, and cells have evolved sophisticated ways to handle it. High specificity in the interactions is key proteins only binding tightly to their correct partners. Think of it like intricate molecular Velcro, only sticking where it's supposed to. And there are noise filters. For example, phosphatases are always active at a low level, cleaning up stray, accidental phosphorylation, so only strong, intentional signals persist.
Filtering out the background noise? Makes sense.
And a really crucial strategy is building intracellular signaling complexes. Sometimes, large scaffold proteins preassemble groups of interacting signaling proteins. They hold all the necessary players together before the signal even arrives. This makes the response super fast, efficient, and prevents the components from wandering off and causing trouble elsewhere.
So like organizing the workers and tools on the assembly line beforehand.
Exactly. Other times, these complexes form dynamically after the signal arrives. Often, they assemble right on the activated receptor itself. The phosphorylated receptor tail can act as a temporary docking platform, or they might assemble on specific modified lipids in the membrane.
And the proteins themselves have special bits for connecting.
Yes, many signaling proteins have these compact modular regions called interaction domains, things like SH2 domains, pH domains. Each type recognizes and binds to a specific structural motif on another protein or lipid. Think of them as standardized connectors like USB ports. They allow proteins to plug into each other in precise ways, building up these complex signaling networks, linear cascades, branching pathways, even large 3D structures.
All right, so signals aren't just on-off binary things. They seem to have different qualities, speed, strength.
Oh, absolutely. The response timing can be incredibly varied. Milliseconds for a nerve firing, hours or even days for a cell deciding its developmental fate. Sensitivity is often extreme. Hormones work at tiny concentrations because of signal amplification. One receptor activates a few molecules, which each activate many more, and so on. A cascade.
A tiny whisper becomes a roar.
A very effective roar. And cells can respond over different ranges of signal strength. Some systems are like a simple light switch. Others, like our vision, can adapt to respond over a huge dynamic range. And responses can be short-lived or persistent, even permanent in the case of cell differentiation.
And cells integrate information, you mentioned.
Yes, they act like tiny computers, combining inputs. They often use coincidence detectors, proteins or complexes that only get fully activated if they receive multiple signals simultaneously, like needing two keys turned at once. It allows for really fine-tuned, context-dependent responses. Plus, a single signal can trigger coordinated changes across many different cellular processes.
Does the speed of the response depend on what the cell needs to achieve?
It does. Really fast responses, seconds, maybe minutes, usually involve modifying proteins that are already there. Just flipping a switch, adding a phosphate, changing shape. Slower responses, minutes to hours, typically require changes in gene expression. Making new proteins from scratch takes time. And importantly, having rapid turnover, constantly making and degrading signaling molecules, allows cells to react quickly when a signal appears, but also shut down quickly when it disappears.
Right, you need to be able to stop the signal too. Now, what about those dramatic shifts? Can a smooth, gradual increase in signal cause a sudden all-or-nothing change in the cell?
Yes, definitely. Signal processing can lead to different response curves. Some are smoothly graded, more signal, proportionally more response. Others are sigmoidal. Not much happens at low signal levels, then the response ramps up steeply. And some are truly all or none, like flipping a digital switch. The cell is either fully off or fully on, with no stable in-between state. This often happens when multiple activation steps are required, creating a high threshold.
What creates these different response shapes, especially the switch-like ones?
Feedback loops are absolutely critical here. Positive feedback is when an output product enhances its own production. A little positive feedback can sharpen a response, making it more sigmoidal. But strong positive feedback can create that all-or-none switch. It can make the system pi-stable, able to exist stably in either the off or on state. Once flipped on, it can stay on even if the initial signal weakens or disappears. It's a form of cellular memory.
Wow, so a temporary signal can slip a permanent switch.
Exactly. Crucial for development. Now, negative feedback is the opposite. The output inhibits its own production. This limits the response, prevents it from getting too strong, and makes the system more stable. If there's a delay in the negative feedback, you can get oscillations. The response goes up, triggers inhibition, response goes down, inhibition fades, response goes up again, like a biological clock.
Like a thermostat.
Sort of, yeah.
Yeah.
And if the negative feedback is fast, it leads to adaptation or desensitization. The cell stops responding to a constant level of signal, but remains sensitive to changes in the signal level. This is how your eyes adapt to different light levels, or your sense of smell adapts to a constant odor. The system adjusts its sensitivity.
That's incredibly sophisticated. Okay, let's dive into those GPCRs, the G protein-coupled receptors. You said they're a huge family.
The largest family of cell surface receptors we have, yeah. Over 800 in humans. They handle signals for sight, smell, taste, plus tons of hormones and neurotransmitters. They all have this characteristic structure, a single protein chain weaving back and forth across the membrane 7 times. Looks like a snake. Often the signal molecule binds deep within that bundle of helices.
And how do they activate that G protein middleman?
When the signal binds, the GPCR changes shape slightly. This lets it grab onto its partner G protein, which is usually nestled nearby on the inner side of the membrane. The G protein has 3 parts: alpha, beta, gamma. The GPCR acts like a GEF. It encourages the alpha subunit to let go of its bound GDP and pick up a fresh GDP. Binding GTP activates the G protein, typically causing the GTP-bound alpha subunit to split off from the beta-gamma pair. Now you have 2 active signaling pieces, the alpha-GTP unit and the beta-gamma complex, which can both interact with downstream targets.
And how does it turn off?
The alpha subunit has its own built-in timer. It eventually hydrolyzes its GTP back to GDP and activating itself. Then it usually rejoins the beta-gamma pair, reforming the inactive G protein, ready for another cycle. Helper proteins called RGS proteins can speed up this inactivation step.
Okay. And you mentioned these GPCRs control cyclic AMP level.
Yes, that's a classic pathway. Some G proteins, called Gs for stimulatory, activate an enzyme called adenyl cyclase, which makes cyclic AMP, CMP from ATP. Other G proteins, called G for inhibitory, block adenyl cyclase. So the balance between Gs and G activity determines the CAMP level inside the cell, and bacterial toxins famously mess with this. Cholera toxin locks Gs in the on state, causing massive CAMP production and fluid loss in the gut. Pertussis toxin locks G off, disrupting its inhibitory signals.
Nasty stuff. So what does CAMP actually do in the cell?
In most animal cells, CAMP's main job is to activate cyclic AMP-dependent protein kinase, or pKa. pKa is normally inactive, but when CMP binds, it releases its active catalytic subunits. These active PK subunits then go phosphorylate specific target proteins, and who those targets are varies a lot between different cell types. That's how the same signal can have different effects. PK can even go into the nucleus and phosphorylate transcription factors like CRD, turning on gene expression. This can translate a short CAMP pulse into a much longer-term change, which is important for things like learning and memory.
Okay, but TAMP isn't the only second messenger from GPCRs, right? You mentioned another pathway.
That's right. Many GPCRs activate a different G protein, GQ, which in turn activates an enzyme called phospholipase C-beta, PLC-beta. PLC-beta sits in the membrane and cuts a specific membrane lipid, PIP2, into two smaller pieces, both of which act as second messengers. One piece is IP3, inositol trisphosphate. It's water-soluble, diffuses into the cytosol, and binds to receptors on the endoplasmic reticulum membrane. These receptors are calcium channels, so IP3 binding triggers a release of stored Ca2+ from the ER into the cytosol.
A calcium flood. And the other piece?
The other piece is diacylglycerol, DAG. It's fatty, so it stays embedded in the plasma membrane. There, it works together with the released calcium to recruit and activate another important kinase called protein kinase C, PKC, which then phosphorylates its own set of target proteins.
So calcium is a really big deal as an internal signal?
Huge. Cells work very hard to keep the normal cytosolic calcium concentration extremely low. There's a massive gradient, much higher calcium outside the cell and inside the ER. So when signals open calcium channels, either from outside or from the ER stores, calcium rushes in, and that spike is a potent signal. There are different types of calcium channels involved, including IP3 receptors and another type called ryanodine receptors.
And you mentioned calcium waves and oscillations.
Yes. Calcium release can actually trigger more calcium release from nearby channels, a positive feedback loop called calcium-induced calcium release, CICR. This can lead to these regenerative calcium waves that propagate across the cell. It's quite dramatic. But then negative feedback kicks in. High calcium can inhibit the channels and pumps work to remove the calcium, leading to these repeated calcium oscillations. The frequency of these spikes often encodes information about the strength of the original stimulus.
How does the cell decode that frequency?
A key player is calmodulin. It's a small, ubiquitous protein that binds calcium. When calcium levels rise, calmodulin binds calcium, changes shape, and can then wrap around and activate other proteins. One important target class is the CAM kinases, CA2 plus calmodulin-dependent protein kinases. CAM kinase 2 is particularly interesting, especially in neurons. It has this amazing property. Once activated by calcium calmodulin, it can phosphorylate itself, which keeps it partially active even after the calcium signal drops back down. This makes it a kind of a molecular memory device, allowing it to integrate signals over time and respond differently to different frequencies of calcium oscillations. It's thought to be critical for learning and memory.
That's incredible. So can you give some real-world sensory examples where GPCRs are central?
Absolutely. Smell is a great one. Olfactory neurons in your nose have specialized cilia packed with different GPCRs olfactory receptors. Each receptor type binds specific odor molecules. Binding activates a specific G protein, GALF, which boosts CAMP levels, opening CAMP-gated ion channels, depolarizing the neuron, and sending a signal to your brain. Smelled something.
And vision works similarly.
Vision is another GPCR classic, but with a twist. Photoreceptor cells, rods for dim light, have a GPCR called rhodopsin. Light acts as the signal, activating rhodopsin. Rhodopsin activates a G protein called transducin, GGM, GTT, but here's the twist. Activated transducin activates an enzyme that breaks down another cyclic nucleotide, cyclic GMP, cGMP. So light causes cGMP levels to drop. This closes cGMP-gated cascade channels in the rod cell membrane. Closing these channels hyperpolarizes the cell, makes it more negative inside, which reduces its signaling to the next neuron in the pathway. So paradoxically, light inhibits the baseline signaling.
Wow, so light turns the signal down.
Effectively, yes.
Good.
And the amplification is just immense. One photon hitting one rhodopsin can lead to the hydrolysis of hundreds of thousands of cGMP molecules and the closing of hundreds of ion channels. That's why we can see single photons. Plus, there are very rapid negative feedback mechanisms involving phosphorylation of rhodopsin, binding of arrestin proteins, and calcium feedback, which allow the system to adapt incredibly quickly to huge changes in light intensity.
Amazing adaptation. What about that gas signal you mentioned earlier, nitric oxide?
Right, nitric oxide, NO. It's unique because it's a gas and can diffuse straight through membranes. It acts locally because it's very short-lived. The classic example is blood vessel dilation. Nerves release acetylcholine onto endothelial cells lining the vessel. This triggers NO synthesis inside those cells. The NO gas diffuses out into the adjacent smooth muscle cells where it activates an enzyme that makes cyclic GMP, cGMP. Elevated cGMP leads to muscle relaxation, and the muscle widens, increasing blood flow. That's how nitroglycerin works for angina. It gets converted to NO. And Viagra works by inhibiting the breakdown of cGMP in smooth muscle.
Okay, so that covers GPCRs. Let's switch gears to the other major class, enzyme-coupled receptors. What's their main mode of action?
The most common type are the receptor tyrosine kinases, RTKs. These usually exist as single units in the membrane. Ligand signal binding typically causes two receptor units to come together. They dimerize. This dimerization brings their cytoplasmic tails, which have tyrosine kinase enzyme activity, close together. They then phosphorylate each other on specific tyrosine residues, trans-autophosphorylation. This does two things. It fully activates their kinase domains, and it creates phosphorylated tyrosine docking sites.
Docking sites for other proteins to bind.
Exactly. Various intracellular signaling proteins contain specific domains, like SH2 or PTB domains, that recognize and bind to these phosphotyrosines. Once docked, these proteins get activated or brought near their targets, relaying the signal downstream. Some docked proteins are enzymes themselves, like phospholipase C-gamma or PI3 kinase. Others are adapter proteins that link the receptor to other signaling molecules.
And this connects to RAS, right? The protein often involved in cancer.
Yes, RAS is a crucial player downstream of most RTKs. It's a small monomeric GTPase, another one of those molecular switches on with GTP, off with GDP. And you're right, mutations that lock RAS in the on state are incredibly common in human cancers, about 30%, driving uncontrolled proliferation. RTKs don't usually activate RAS directly. They recruit adapter proteins, like GRP2, which bind to the phosphotyrosines. GRP2 then recruits a RAS GEF, like SOS, which activates RAS by promoting GTP binding.
So the RTK acts like a matchmaker to get RAS activated. What does RAS do then?
Activated RAS kicks off a major signaling cascade called the MAP kinase module. MAP stands for mitogen-activated protein kinase. It's typically a 3-kinase relay. RAS activates RAF, MAP kinase, or milky kinase. RAF activates MEK, MAP kinase, and MEK activates ERK, MAP kinase, or MAPKK. Activated ERK then phosphorylates loads of different target proteins, including transcription factors in the nucleus, leading to changes in gene expression, often promoting cell growth and division. Interestingly, the duration of MAP kinase signaling can influence the outcome. A short pulse might lead to division, while sustained signaling could trigger differentiation.
And scaffolds are important here, too, to keep things straight.
Very important. Scaffold proteins often hold the RAF-MEK-ERK kinases together, ensuring the signal flows correctly and preventing unwanted activation of parallel MAP kinase pathways that might use similar components.
Okay. You also mentioned pathways controlling cell shape and movement.
Yes, that's largely the domain of the Rho family GTPases like Rho, RAC, and CDC42. These are other small GTPases similar to RAS. They relay signals, often from RTKs or other receptors, to regulate the cell's internal skeleton, the actin filaments, and microtubules. By controlling the cytoskeleton, they control cell shape, polarity, movement, adhesion, critical stuff. For instance, signals can activate Rho, leading to changes in the actin cytoskeleton that cause a migrating cell or growing nerve axon to retract.
So proteins get phosphorylated, but lipids can too, you said?
Absolutely. PI3 kinase is a key enzyme here. Often activated by RTKs, it phosphorylates inositol phospholipids in the plasma membrane, specifically at the 3 position of the inositol ring. This creates specific lipid docking sites, different from the ones made by PLC. A key lipid product is PIP3. And just like phosphorylation, this lipid modification is reversible. An important phosphatase called PTN removes that phosphate. Loss of PTN function is another common event in cancer, leading to excessive survival signaling.
And what gets recruited to these lipid docking sites?
A crucial pathway involves the recruitment of protein kinases that have pH domains, which bind to these specific phosphorylated lipids like PIP3. This brings kinases like ACT, also called protein kinase B or PKB, and PDK1 to the membrane, where ACT gets activated. Activated ACT is a major promoter of cell survival and growth. It phosphorylates many targets, including proteins involved in apoptosis, effectively putting the brakes on cell death.
So another anti-cancer drug target, presumably?
Definitely. And ACT also feeds into regulating cell growth through a complex called MTORC1. MTOR integrates signals about growth factors via ACT and nutrient availability, like amino acids, to control protein synthesis and cell growth. It's a central hub for growth control.
It seems like these pathways, RTKs and GPCRs, while starting differently, might activate some of the same internal machinery.
They definitely do. There's significant crosstalk and convergence. Both receptor types can activate PLC, for instance, PLC beta for GPCRs, PLC gamma for RTKs. Both can influence MAP kinase pathways. This allows cells to integrate signals coming through different receptor types, fine-tuning the overall response.
What about receptors that need tyrosine phosphorylation but don't have their own kinase domain?
Good point. Those receptors rely on recruiting cytoplasmic tyrosine kinases. These are kinases like the Slancer family, or FA, focal adhesion kinase, that are not part of the receptor but associate with it. When the receptor binds its ligand, it activates these associated cytoplasmic kinases, which then phosphorylate the receptor itself and other downstream targets. Integrins, which mediate cell adhesion, work this way, often activating kinase.
And the JAK-STAT pathway fits here too.
Exactly. Receptors for cytokines and some hormones often lack intrinsic kinase activity, but are tightly associated with cytoplasmic tyrosine kinases called Janus kinases, JAKs. Ligand binding brings the receptors and their JAKs together. The JAKs phosphorylate each other, then phosphorylate the receptor tails. These phosphotyrosines become docking sites for proteins called STATs, signal transducers and activators of transcription. STATs dock, get phosphorylated by the JAKs, then detach, dimerize, head straight to the nucleus, and directly activate gene transcription. It's a very rapid pathway from receptor to gene activation.
Very direct. What about the TGF-beta family? They're important in development too.
Yes, the TGF-beta superfamily includes many secreted signaling proteins crucial for development. Their receptors are different again. They are receptor serine kinases. Ligand binding brings together two types of these receptor kinases, type 1 and type 2. The type 2 receptor phosphorylates and activates the type I receptor. The activated type I receptor then directly binds and phosphorylates intracellular signaling proteins called SMADs. Phosphorylated SMADs team up with another SMAD, SMAD4, move into the nucleus and regulate the transcription of target genes, another direct route to the nucleus.
Okay, speaking of direct routes, some pathways seem to use regulated protein cutting, proteolysis, to activate signals.
That's right. Regulated proteolysis is another key strategy, especially for controlling latent transcription factors that are just waiting to be unleashed. The Notch pathway is a prime example, vital for cell fate decisions during development. A classic case is lateral inhibition, a cell telling its neighbors not to adopt the same fate. The Notch receptor sits in the membrane. When it binds its ligand, delta or serrate, on an adjacent cell, it gets cleaved by proteases. A part of the Notch cytoplasmic tail is released, travels to the nucleus, and directly participates in activating Notch target genes. It's incredibly direct.
And there's a link to Alzheimer's disease here.
Yeah, intriguingly. One of the proteases involved in cleaving Notch, called gamma secretase, contains a protein called presenilin. Mutations in presenilin genes are a major cause of early-onset familial Alzheimer's, related to how it processes another protein involved in amyloid plaques. A fascinating, if unfortunate, connection.
Wow. What other pathways use this protein-cutting strategy?
The WNT beta-catenin pathway is another huge one in development. WNT proteins are secreted signals. The core of the pathway is beta-catenin. Normally, in the absence of a WNT signal, beta-catenin is constantly being tagged for destruction by a large protein complex in the cytoplasm. But when WNT binds its receptors, frizzled and LRP, this destruction complex gets inactivated. Beta-catenin is no longer degraded, so it accumulates, moves into the nucleus, and partners with transcription factors to switch on WNT target genes, often related to proliferation. And clinically, mutations disabling the destruction complex, especially in a protein called APC, are found in the vast majority of colon cancers, leading to constant WNT signaling and growth.
So glycan degradation leads to activation. What about hedgehog?
The hedgehog pathway, also crucial in development, works a bit differently, but also involves regulating protein processing or stability. Uniquely, many components of the vertebrate hedgehog pathway are localized to the primary cilium, this little antenna-like structure on many cells. Without hedgehog signal, its receptor patch keeps another protein, smoothened, suppressed. When hedgehog binds patched, smoothened becomes active, moves into the cilium, and initiates a cascade that ultimately prevents the cleavage of lunkily transcription factors into an inactive form. The full-length active gly proteins then go to the nucleus to activate target genes. Aberrant hedgehog signaling is linked to basal cell carcinoma.
And NF-kappa B,
Yeah.
that's involved in inflammation, right?
Yes, the NF-kappa B pathway is central to immune and inflammatory responses. NF-kappa B proteins are transcription factors normally held inactive in the cytoplasm, bound to inhibitor proteins called EKB. Various signals, like inflammatory cytokines or pathogen components, trigger a cascade that leads to the phosphorylation and subsequent degradation of EKB. This frees NF-kappa to rush into the nucleus and turn on hundreds of genes involved in inflammation and immunity. It also has a built-in negative feedback loop. NF-kappa B turns on the gene for its own inhibitor, EKP, which helps shut the response down again, sometimes leading to oscillations in activity.
Okay, we've covered signals hitting the outside and triggering internal cascades or direct routes. What about signals that just walk right in?
Those are typically small, hydrophobic molecules that can diffuse across the plasma membrane. Things like steroid hormones, cortisol, estrogen, testosterone, thyroid hormones, vitamin D, retinoids. They bind to intracellular receptor proteins known as nuclear receptors. These receptors are often already in the nucleus or move there after binding their ligand. They are themselves transcription regulators. Ligand binding usually causes a shape change, making the receptor shed inhibitory proteins and recruit coactivator proteins, allowing it to bind to specific DNA sequences and regulate gene transcription directly.
Very efficient. Now, slightly different topic, but related to internal regulation, circadian clocks.
Right, circadian clocks. These are the internal timekeepers that regulate our daily rhythms, sleep-wake cycles, hormone release, metabolism. At their core, these clocks rely on delayed negative feedback loops in gene expression. Basically, clock genes turn on and produce proteins. These proteins accumulate, and after a delay, they act to inhibit the transcription of their own genes. As the proteins degrade, the inhibition lifts, the genes turn back on, and the cycle starts again, typically taking about 24 hours. What's really amazing is the clock in cyanobacteria. It's just 3 proteins, Chi, Chi B, Chi C, that even in a test tube with ATP can run through a 24-hour cycle of Chi C phosphorylation and dephosphorylation. Pure protein-based timekeeping.
Incredible. We spend a lot of time on animal cells. How do plants handle signaling? Did they invent completely different ways?
It's a great comparison because plants and animals evolved multicellularity independently, so there are similarities but also striking differences. Both use some common tools, calcium signaling, NO, protein kinases, small GTpases, but plants lack many of the big animal players. No GPCRs in the same way, no RTKs, no nuclear receptors for steroids, no Notch, Wnt, Hedgehog, JAK-STAT, and they don't seem to use cyclic AMP.
So they built their communication systems from different parts lists?
Largely, yes.
Yeah.
For instance, the biggest family of cell surface receptors in plants are receptor seronephronine kinases, often with leucine-rich repeats, LRRs, on the outside for ligand binding. Animals use these too, but RTKs are more dominant for growth factor signaling. Plant hormones are also quite different.
Like ethylene, the fruit ripening gas.
Exactly. Ethylene is a gaseous hormone. Its receptors are in the ER membrane, and bizarrely, they are active when empty, constantly signaling to keep a downstream transcription factor, EIN3, targeted for degradation. When ethylene binds, it inactivates the receptors. This stops EIN3 degradation, allowing it to accumulate in the nucleus and switch on ethylene-responsive genes for ripening, leaf drop, etc. It's completely counterintuitive compared to most animal receptors.
Off means on and on means off. Wild. What about auxin?
Auxin is another crucial plant hormone, controlling growth, root formation, responses to light and gravity. Its receptors are actually part of the machinery that targets specific repressor proteins, auxiase, for degradation. Auxin binding promotes the degradation of these repressors, thereby allowing auxin-responsive genes to be expressed. But perhaps the most unique thing about auxin is its polar transport. Plants precisely control auxin flow by positioning specific influx and efflux carrier proteins asymmetrically in their cell membranes. This directed flow of auxin guides growth patterns, like ensuring roots grow down.
So transport is the signal direction.
In a way, yes, the pattern of transport is critical information. And finally, light sensing is huge for plants. They have photoreceptors like phytochromes, detecting red light, often acting as kinases that move to the nucleus, and cryptochromes, detecting blue light, also involved in circadian clocks.
Wow. Okay, we have covered an immense amount of ground here. From the basic idea of cells needing to talk through these incredibly complex internal pathways with switches and feedback loops, right up to the distinct strategies used by plants, the elegance is just staggering.
It really is. Understanding this intricate web of communication is so fundamental. It's the basis for understanding development, disease, physiology, everything. And advances here are constantly leading to new therapeutic ideas, but also just a deeper appreciation for how life works at this microscopic level.
Absolutely. Well, thank you for joining us on this deep dive into the world of cell signaling. We hope you listening picked up some key insights, maybe had a few aha moments. Hopefully it sparked even more curiosity. And here's something to think about. We talked about adaptation, how cells adjust their sensitivity to constant signals. If our cells do that constantly, how much are our own perceptions, our moods, our responses to the world around us shaped by similar ongoing adaptation processes that we're completely unaware of? Something to ponder. Thanks for listening.